what are we doing here today? Well, what we're doing today is normally in a Biology 101 class, we would have a lecture associated with the class. However, you are online students, and therefore, typically, there is no lecture. What we're trying to do with these presentations each week is provide a lecture opportunity to online students. And the way this will be structured is as follows. There will be a 40 to 60 minute presentation followed by 30 minutes for questions. Now keep in mind, when I'm doing these, I'm more than happy if you would like to ask questions along the way. Just raise your hand as you have before while we were figuring out the voice things, and I will answer your questions for you. So don't hesitate. That being said, we kind of know why we're all here. Let's go ahead and talk about why genetics is important to your understanding of biology for a moment. Genetics is important to your understanding of biology because all living things have a genetic basis. All living things have a, oh, we got a new question there, sorry. All living things have DNA, and that DNA codes for what they look like. And so there's still music playing. All those people should be muted. I'm going to try to remute. Everybody here. All right. Is that music still in the background? I'm going to try to mute that again. How about now? Mary, that's coming from your computer. That's actually on your end, because when I mute you, the music goes away. So that's something that you have in your computer in the background, probably. All right, so back to where we were. The reason why, and also just to let you know, this will be available online later, and so that copy will obviously not have the music in it if that's the case. So if that ends up being a problem, you're welcome to both attend and review the video later. So back to why genetics is important for studying biology. The biology is the study of life, and all living things have a genetic basis. And so that's why we're going to take some time today talking about genetics. Let's switch gears for a moment, and I'll kind of give you an overview of what we will be covering. And we kind of already talked about what we'll be doing today. We will then go on to why genetics is important for the studying of biology, which we already covered. And then here's kind of the overview of what we'll be talking about. We'll be talking about what is a phenotype, what is a genotype, chromosomes, genes, and alleles, and how they're all connected. We will then link the genotype to the phenotype and talk about classic Mendelian genetics. This includes subjects of homozygous and heterozygous alleles. We'll talk about Punnett squares using eye colors as an example, and we'll move over to the whiteboard to do some of that. We'll talk about pedigrees as well as polygenetic inheritance and how phenotype is really a function of both genetics and the environment. And so let's go ahead and dive right in with our discussion of what is a phenotype and genotype. So back to me now. The phenotype is how something looks. And so really, a phenotype can be any measurable quality of an organism. And so one of the obvious phenotypes that people talk about a lot is height. or hair color, or skin color, or eye color. All of these are measurable things. But don't just think that this is limited to things that are easily observed with your eyes. For instance, if I asked you all to run for 10 minutes and then recorded your heart rate after that 10 minutes, that would be a phenotype. It would be a phenotype of heart rate after exercise. And so phenotypes can be very varied, including things like physiology, anatomy, behavior, pretty much anything you can imagine. And so if that's phenotype, we can also ask, what is genotype? Well, genotype is genetics. It is the genetic components that underlie that trait or that phenotype that you're measuring. And a phenotype will be influenced by the underlying genotype. So a specific gene codes for a specific phenotype. Let's go ahead and link this together a little bit by looking at chromosomes, genes, and alleles. And so we're going to move over to the whiteboard here. And a chromosome is a bundle of DNA. And you as a human have 46 chromosomes. And these chromosomes come in pairs. And the way this works is you have 2 chromosomes, and one of those came from your mother, and one of those came from your father. And the reason is, if you think about it, sperm and eggs each have 23 chromosomes. So when a sperm and egg unite, that gives you a total of 46. And each of these chromosomes are complementary to each other, meaning they're similar. They have a lot of the same information, but they can have different variants of that information. So on a chromosome, there is a location, and that location on this chromosome may have a specific gene. A gene is a series of the A's, T's, C's, and G's that you learned about. If you read the chapter, these were all over your book. And when we divide these into groups of 3, they code for amino acids. And if we put enough of these together, we end up with a gene that codes for something. And maybe it's something that says makes a protein that ends up resulting in changing the color of something. The way this works then is each of these are complementary, but they can be different. For a single location, for a single gene, we can have different alleles. And what those alleles are is they represent that genotype that we were talking about. So let's go ahead and link that genotype to the phenotype, and we're going to use the example of eye color. Okay, so in people, we know that we can have blue eyes and brown eyes. Those blue eyes and brown eyes are a phenotype. They're something we can measure about the organism. And so, obviously we know there's hazel green and some variations. We're going to simplify all of this for our exercise today. These phenotypes have an underlying genotype. Each of them has an allele from, remember, we had 2 genes for eye color. Blue eyes have 2 alleles, and we're going to call these little b that represent blue. Brown eyes have 2 as well. And here's where things get a little bit interesting, because the big B codes for brown, and so 2 big B's would definitely be brown. However, a big B and a little b is also brown. So what does this mean? Well, in this case, brown eyes, or the big B, are dominant to blue eyes, or little b, which is the recessive trait. When the alleles are the same, this is called homozygous. So big B is homozygous, little b is homozygous. But little b is known as homozygous recessive, and big B is known as homozygous dominant. Heterozygous traits are one where we have two different alleles for that trait. And in this case, the big B, little b are two different ones, obviously, so that would be heterozygous. And so when you look around the world, you can look at people, and you can know for certain that blue-eyed people have little b, but that brown-eyed people could be homozygous or heterozygous. So let's go ahead and take this a step further now, and let's look at directly how mating events might translate to eye color. So let's say you were wondering if you have blue eyes and your partner has blue eyes. We'll take a really simple example first. What would be the odds of your children having a certain color eye? So what we do is we know that you have two chromosomes, and each of those has one allele. But when you make your sperm and eggs, you actually reduce this number to one that is in the sperm. So you only can pass on one of these. And so in the case of somebody who has blue eyes, we would say blue eyes is little b is another possibility. And if their partner has blue eyes, little b is a possibility again. And so what we can do is draw what's known as a Punnett Square. And in each of these squares represents the chance of getting these two alleles combined. And so, for instance, little b here would look like this, because they could get this allele or this one. And so in this case, these people, because they have blue eyes, they could only have blue-eyed children, because this is the recessive trait, and so we know their genotype is this. Let's go ahead and use an example that might make it a little bit more clear. Let's get rid of this, and let's use two heterozygous individuals. So we erase there. And let's just use another, let's use a color here to denote this. Let's say that big B's we're now going to put in, actually, let's put them in blue to make it easier. All right, so let's get rid of them in black. Here is blue, and this one stays black, and this one is blue. So if we looked at a person's chromosomes, and they were heterozygous, it means that one chromosome would have a big B, and the other chromosome would have the little b. Like that, so on this second chromosome. But they are only going to take that gene from one of the chromosomes during the reproductive event to put it in the sperm or the eggs. So there is a 50% chance you end up with big B, 50% chance that that sperm or egg has the little b. And if they mate with a person who is also big B, little b, there's a 50% chance that that person would give them each of these alleles. So think about this for a second, because what this means is that somebody who has brown eyes could still be a carrier for the blue trait. What do I mean by that? Since blue is recessive, an individual with the blue eyes will end up, because it's recessive, an individual will not have blue eyes. The brown is dominant. And so if you've ever heard of something skipping generations, or, you know, somebody having different color eyes than their parents or something like that, this is oftentimes the reason. Because when we draw out this Punnett Square, what we'll see is that this person could be homozygous dominant, or big B, big B. They could be big B, little b. here's another chance where they could be big B, little b. And here's the last one where they're little b, little b. So what we have then is a 25% chance if two people with brown eyes who are heterozygous mate, that the genotype of their offspring is homozygous dominant, or in other words, big B, big B. There's a 50% chance when they mate that the genotype of their offspring is heterozygous, or big B, little b. However, this also, remember, produces brown, because the effects of this allele dominate that one. And so in this case, there would be a 75% chance that this couple would have a brown-eyed child. There is a 25% chance that this couple would have a blue-eyed child, or a 25% chance the genotype would be little b, little b. And so that is kind of the gist of Mendelian genetics that are under there. Why don't I give you guys a moment to ask questions for a second. If there are no questions, we can just move on forward. Okay, it looks like you did not have questions. So let's continue with this, and I'd like to point out something interesting known as a Punnett Square. And so what the Punnett Square is going to show us, sorry, not Punnett Square, pedigrees is what we're moving on to. The PowerPoint is not responding. There we go. Let's go ahead and show you the pedigree. We need... All right. So this is an example of a pedigree. And what you can see here, if you can see my mouse moving around there, is the same sorts of setups that we had before. In this case, we're talking about albinism, or something being albino. And if they have a large A, they will be normal. But if they have, I'm sorry, I think I'm saying that backward. If they have two little a's, they will be albino. But if they have a large A, that's dominant, and they will not be. And so what you can see here is with a pedigree, you can actually use this to help determine what you can expect from your offspring, and it helps to have information from generations in the past. So if this wasn't an albino example here, and it was the eye color again, what you might expect was, even though you might have two people with brown eyes, if you know that one of those people with brown eyes had a parent with blue eyes, you know they must have at least one little b allele. And so by getting as much information as you can about your family, you can make predictions about the likelihood, using Punnett squares then, of a offspring having a certain characteristic. And this is what genetic counselors used to do a lot of. They would use these sorts of techniques to help determine what likelihoods of diseases and things like that are. Nowadays, we have other ways we do that. There are several hormone tests, but there's also direct genetic testing for some issues like this. And so even though this is a very basic way to determine it, it is still used. So let's go ahead and move forward then to our next portion. And we just covered chromosomes, genes, and alleles. We showed how genotype and phenotype were related in classic Mendelian genetics, and we talked about these homozygous, heterozygous alleles, and we showed you a Punnett square with eye colors as an example, and we talked about pedigrees. Now let's move on to polygenetic inheritance. And polygenetic inheritance is the way that actually most of the traits that you see in the world are set up. So let's go back to showing you me here. And so what do I mean by that? Polygenetic means that a trait is determined by many genes. So not just one gene determines a trait. And so what this means is can be a lot of variability in offspring. And so if we take a look at height again, and let's say that height was controlled by many genes, but all these genes still follow classic Mendelian genetics. And so what we have would be a big T for tall and a little t for short. And so tall could be big T, little t, or big T, big T. Short could only be little t, little t. And so here's something to think about. Have you ever known somebody, well, let's put this a different way. If you were to guess how tall somebody's children would be, you might look at their parents and you might say, well, their parents are tall, so we presume their children will be short. Or the parents are short, so we presume the children will be short. Why? Because we intuitively know. We know that genetics are linked to the phenotype. However, that doesn't always happen, right? We can all probably think of examples of people we know where the parents are tall but they have short children, or the parents are short but they have tall children. And so it's possible to get variation. Offspring do not directly 100% resemble their parents. And so if we take our example of height here, let's imagine that this was controlled by five genes. And so there's going to be 1, 2, 3, 4, 5 genes that code for height. And each of these is going to have the possibility of a big T or a little t. So this would be, for instance, T1, T2, T3, and so on. So let's imagine we had an individual who had this genotype. Okay. In that case, if you can't see that very well, what I drew there is they were heterozygous at every loci. So for all 5 of these genes, they were heterozygous. And let's say they mate with somebody who is also heterozygous at every locus, or loci, sorry. Well, this presents an interesting scenario. And this is one where we might initially assume that these two people are both tall, right? In every 5 of these locations, they had the tall gene. And so what this means is each of these genes is saying be a little bit taller, be a little bit taller, be a little bit taller, be a little bit taller. They're working together additively. The more tall genes you have, the taller you will be. And both these people are very tall. But when these people mate, consider the fact that they're heterozygous. Even though they're tall, each of them has some short genes. So what this means is that there's a chance they will have very tall offspring, but there's also a chance that they could have short offspring. Because let's say that they both happen to pass along, when each of these, they send one of the two a bunch of the short alleles. If in those cases they both tend to send short alleles, that offspring will have more alleles for being short and will end up shorter. And so this is why height makes a good example of a trait with polygenetic inheritance, where you have a trait that is determined by multiple genes. And as I mentioned before, many traits, in fact, most are determined by multiple genes. And this is just one example where those genes working together are, in fact, additive. So let's take a graph of how this would look and consider how this plays out in the real world. Because we're going to draw here what's called a mid-parent offspring regression. And when we draw this, this is mid-parent height. And what that means is we take mom's height, we take dad's height, we add them together, divide by 2. That gives us our average. And so here we have short, and here we have tall. And over here, what we're going to show is offspring height. Offspring height. And here we have short, and here we have tall. And so take a minute to think about this. We talked about it in words before, but we didn't draw it. How would you expect this to look? Well, if both somebody's parents are short, what would their offspring look like? And you're probably thinking, yeah, well, they're going to be short then. If both their parents are tall, well, their offspring are probably going to be tall over here. What about if they're medium height? Well, we would presume that most of them end up as medium height. And so what we end up with here is a line that shows the genetic relationship between a parent's height and offspring height. If parent's height is highly related to offspring height, it means that trait is determined very genetically, very simply. But we know it's not always so simple. Because what can happen is that somebody who's medium height has both tall genes and short genes and can maybe make somebody who's a little bit taller or somebody who's a little bit shorter. So we end up with variation around that mean. And same thing down here. They could have somebody who's very short, taller, like this. We have variation around those. And so that would be how you have tall parents who make short children and vice versa. On average, they're more likely to be taller, and I should maybe draw these a little longer, but there's the possibility for them to still be relatively short. But this isn't the end of the story, because everything, your traits, I should say, are not just determined by the environment. Your phenotype is determined by genetics and the environment. And so let's get rid of our genetics, our genotypes over here. And let me show you a very important equation to the understanding of an organism's phenotype. Oh, I seem to be stuck over here. There we go. And what that is phenotype equals genetics plus environmental effects. And what do we mean by that? It means that phenotype, how something looks, or any measurable quality of that organism, will be a function of both genetics and the environment. Keep in mind that this is going to be true for almost every trait you can think of. And let's use our height example again. And what's going on in that example is that we said, well, taller parents should have taller offspring because genetically they're more likely to. They have more tall alleles, more tall alleles getting passed on. They're more likely to be tall. But are all parents really good at raising their children? Maybe not. Maybe some parents really suck. And maybe these parents don't do a good job of feeding their kids. And so even though this child has a bunch of genes to make it tall, what might happen if while it's growing it never gets enough nutrients? It never gets enough food. Well, there's the potential that its growth could be stunted. And that would be an environmental effect. So even though genetically you're inclined to be tall, because you didn't get enough food, you ended up shorter. And so you can think about this for a great number of traits. Let's think about something like skin color. Skin color, genetically, you may have a certain skin color, but in the case of people with lighter skin, if they go out in the sun, environmentally, it becomes darker. And so there's a wide variety of traits that are determined by genetics and the environment. you may have heard this expressed in another way at some point as an argument of nature versus nurture. Or in other words, why does somebody act the way they do? Is it because they were born a certain way or because of the way they were raised? The nature being the way they were born, the nurture being how they were raised. And the truth is, there really is no answer. For any given behavior, there's going to be a certain amount of genetics that determine how a person acts, and there's going to be a certain amount of the way they were raised or environment that determines how they act. And so it's really important to consider both these things at the same time. Let's take another example of this and talk about clones. So in Chapter 15, you read about clones, and clones are basically genetically identical copies of an offspring. So actually, twins are a form of clones. We think about that term a lot of times as being kind of organisms that were intentionally cloned in the laboratory and things like that. But really, two twins that are born, while they may look the same when they're born, are then subjected to different environments. And so what that means is these two twins have the same genetics, and that may make them similar, but there are going to be lots of differences based on the different experiences they have in their life. And so you may know some people who are twins, and yeah, they may have lots of similarities, but they're also going to have a great many differences as well. This is why in movies, when you hear about them, oh, I'm going to clone somebody, and they have this person born, and they're just like the original person, that would almost never happen because there's going to be differences in the way things are raised and how they ultimately end up acting. And so I'm using behavior as an example there, but you could also consider something like the size of an organism, right? Something could be genetically inclined to be a certain size, but if you eat a lot, you'll get bigger. If you don't eat enough, you'll get smaller. And so we still see differences between twins. So I've been looking for questions as we've been going. Do either of you have any other questions at this point before we move on into some nuances? This would be a good opportunity for you to ask something, and I'm going to dive under my desk here for a moment. My mouse fell off while I wait for you to type any up if you have them, or if you'd like, I can give you the microphone to ask them. We don't have a big enough class here that could be possible. All right, so it looks like there are no questions at this time. Let's go ahead and talk then about some specific examples of deviations from some of these processes. We already talked about the polygenetic inheritance. There are also things such as, let me find the slide here to show you guys. Where you can have more than one allele for a trait. And so we can take that example of, say, the A, B, and O blood groups. And so what we've shown you so far is kind of what's normal, the Mendelian genetics. And then we showed you some polygenetic traits polygenetic traits that had many genes. But we can also have different alleles. And a great example of this is the A, B, and O blood groups. So A, B, and O. Now keep in mind, we still have our chromosomes, and each of these chromosomes can have an A, B, or an O. That's how that's set up. And so, let me get my questions tab back. Oh, okay, I've got a question there. I didn't see that. Let's go ahead, and I'm trying to expand your question. I see. And when they were born, they each had their own sac and placenta. Those would be twins that are non-identical. So the question is that if offspring are born and they each have their own sac and placenta, when that's the case, the twins are non-identical, and they are only as related to each other as a regular brother and sister are. And so it would be the same sort of situation as if a brother was born 1 year and then 9 months later a sister was born, or vice versa, two brothers, two sisters. They will not share as much genetics. They will not be genetically identical. So were there any other questions? Okay. Sorry, I'm trying to get this to expand a little bit, and the questions are being difficult. If you have a question again, just go ahead and raise your hand. This seems to be being tricky here. There we go. So let's move on to the A, B, and O blood groups. So in this case, we have 3 different alleles for the gene of blood type, but there is still the same sort of each chromosome in the sperm or the egg combined. And so what you can end up with, instead of having, say, big B, little b, is you can end up with a B, A, O, A, B, 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 O, and O, O is how that gets set up. And the reason for that is that there are, at this location, it's not just 1 or 2 things, there's the possibility of 3. Each person still only has 2 alleles, but there's 3 forms of those alleles. And so you end up with all these possible blood types. And the way this works is AB is, as you may have heard it before, is AB. O doesn't really code for anything. And so you also have type A here. BB and BO are also B. And the OO is your chance to get the O type. And so that's a variation on the standard Mendelian situation that we're talking about. And so at this point, are there any other questions? Or we can go ahead and start to wrap things up here. All right, you can either ask it out loud. I'm going to go ahead and give you guys both mic control here in case you'd like to speak your answers.
No, I don't have any questions.
Okay, and was that okay? You don't either. So is there anything else you'd like clarification on then?
No.
Alrighty, well then we will call that a wrap for today. I apologize for those technical difficulties in the beginning. We got those worked out, and we will be doing this again for the rest of the semester.
Thank you.
Yes, you're Yep, you're very welcome. I hope that helped you understand these concepts a little bit more.
Yeah, that helped a lot. Thanks.
Great, great, good to hear.
Same here. Thank you.
Yep, and I'll be kicking you guys out now. So.
Okay, bye-bye.
Yep, bye-bye.